So in this video we're going to have a discussion about the principle of osmosis, which is a new word, and this is the definition. It's a special type of diffusion which we've seen before, so we should know that molecules will always move by diffusion from an area where there is a higher concentration to an area where there is a lower concentration. And because this is biology and we're talking about movement into and out of cells, we have to think about this idea, the semi-permeable membrane. All cells have them. We've got one here in the diagram at the bottom. And semi-permeable just means that, as you can see in the diagram, there are tiny little holes through which things can move. The membrane is semi-permeable. It lets certain things through, small molecules like water, but larger things are not going to be able to pass through, such as the sugar molecules. And osmosis is new, but it's pretty easy to understand. That we've got two solutions. Solution A is pure water. That's the highest concentration of water molecules you can get, and it has no sugar molecules in it, so it's the lowest concentration of sugar that you could possibly get. And on the other side of the semi-permeable membrane here, we've got a solution that's got some water molecules in, but it's also got some sucrose molecules in. And so this has a lower water concentration in comparison to the solution on the left, and it's got a higher sugar concentration in comparison to the solution on the left. But as far as you're concerned, just think about water. High concentration of water on the left, lower concentration of water on the right. And so water molecules are moving in both directions, and you see on the black arrows that moves from left to right and right to left because they're moving randomly. But because there's more of them this side than there is this side, more of them over time will move in the direction of the pink arrow until eventually this solution starts to become a bit weaker, and you can see that the level on that side would rise, and because the water molecules are moving out of this solution, that level is going to drop. What we need to do is put this in the context of cells. If we look at osmosis in animal cells, here we've got some red blood cells, and this diagram describes what happens when you put them in different types of solutions. So for example, if you put some red blood cells into fresh water, what we describe as a hypertonic, and we'll just talk about that in a second, these cells will swell up and eventually they will burst, just like balloons, because remember, an animal cell only has a membrane around the outside. If you put them into seawater, which has got lots of salt in it, so less water now, more salt, what happens is these cells will actually lose water, they shrink, and the posh word for that is crenate, and they just will scrunch up, a bit like sucking all the air out of a balloon. If you put them into an isotonic solution, there's no change because water isn't moving into or out of these cells. Okay, well, how can that be, and what do these terms mean? So I've just drawn out these key terms here for you to have a look at. So if we consider a red blood cell, a red blood cell has a certain amount of water in it, and it also has other things dissolved in it, like sugars and amino acids. And any solution which has exactly the same water concentration as our cell contents is going to be described as isotonic, and you might have heard that with drinks before. Any solution which has more water in it, so we're going in this direction now towards pure water, is described as being hypertonic, and we saw this in that idea at the top. So a hypertonic solution just up here, like fresh water, and the red blood cells swelled up. And the reason for that is because in this case, the higher water concentration is outside. So water's going to be moving into these cells. Okay, we can draw that on. Water's going to be moving into these cells from the outside, where there's a high concentration of water, to the inside, where there's a lower concentration of water. At the other end of the scale, as we move towards sort of, for example, a pure sugar solution like golden syrup, then what's going to happen as you go this way is that you end up having a hypertonic solution outside. And that's what's happened just here, where these guys are in some seawater, or if we think about the solution that was on the left-hand side, as soon as you start adding sugar in, there's less water to be able to move, and so in this case, water actually starts to move out of these cells because there's a higher concentration inside than there was outside. So this is a lower concentration of water. And so membranes allow water to move in and out. It just depends on where the higher and the lower concentrations of water are. If it's higher outside, water will move in. If it's higher inside, water will move out. If we think about osmosis in plant cells, if we put some plant cells, like some potato cells for example, into an isotonic solution, because the concentration of water outside and inside is exactly the same, what you can see is that water will move out and water will move in. The effect is that there is no net change in this cell, it just remains as it was before. But interestingly, if we take that same cell and put it into a hypotonic solution, remember the hypo bit means that there is now more water outside, the higher concentration is here, it's outside of the cell. What happens is water will move in, and just like a balloon and those red blood cells, they start to swell up. Now this cell, unlike the red blood cell, won't burst, and that's simply because around the outside of a plant cell you have a cell wall, and the cell wall is made of cellulose, it's very tough, and just like putting a balloon into a cardboard box, you can inflate the balloon, but ultimately once it reaches the dimensions of the cardboard box stops it from expanding anymore and prevents it from bursting. If you take those cells and go the other way and put them into a hypertonic solution, so one that's got lots of sugar in but not very much water, so it's a lower concentration of water now outside, you can see that water is going to leave the cell, and that's where it becomes what we call plasmolysed, and this means that the membrane starts to pull away from the cell wall and almost deflates inside its own cell wall. So a turgid cell is one that is absolutely packed full of water, it's very, very rigid and strong. A plasmolysed cell is one which has lots of the water taken out and it sort of collapses in on itself. And this is why plants start to wilt, and this is why plants like leaf cells can hold their shape and keep leaves nice and flat, because they're quite rigid. Let's look at that in the context of an experiment. Here's a nice graph that we can have a look at. So in this graph what we've done is we've put some potato cells, for example, into different solutions of sucrose, different sugar solutions, and we've measured the change in mass. Because each piece of potato had a different mass to start with, we've looked at percentage rather than the grams that they've added or taken away. And if you look at this graph, it changes from being up here where it's a positive change in their mass, which means that they've gained mass, to a negative change where they've actually lost mass. And in between, at this point here, the change of mass was zero, it stayed the same. And that's because this point is where we have an isotonic solution. That tells us the concentration of the water outside was the same as the concentration inside the cells, and therefore the net movement was zero and the mass didn't change. Up here, where we have a positive change, this solution, so anything this side of the green line, must be hypotonic. The solution outside had a higher concentration of water, so water's moved into the cells, and the cells have gained mass. You can see here the mass is above the zero line. This side of the line, where the line shows you that the cells have all lost mass, well, this must have been hypertonic. The concentration of water outside was lower, it was higher inside, so it's moved from being inside to being outside, and that's caused the mass to decrease. And what you can see is that the bigger the difference between the inside and the outside, the more or the more water was moving. So the bigger the gain in mass, or the bigger the loss in mass.